Hello, welcome to lesson number 22, and this lesson covers the topic contamination prevention and control, and this is actually the last lesson in this particular unit under the heading of quality assurance aspects. So at the end of this lesson, the topics that we'll have covered include the definitions associated with contamination prevention and control, the different types of contamination. We give a few examples of different types, how to prevent contamination, as well as discussing clean rooms and regulations around contamination prevention and control. So as an introduction, any manufacturing environment is critical for product quality, and these include things like the lighting that's used in the manufacturing process, as well as the temperature and humidity, as some products are sensitive to both of those things. The air movement within a manufacturing site needs to be regulated and monitored and controlled at all times to prevent dust or particles traveling between containment areas. Microbial contamination and particulate contamination are two things that you need to be very much aware of during manufacturing. Uncontrolled environment can lead to product degradation, product contamination, loss of product, and obviously loss of profit. So the two key definitions that I just wanted to highlight are contamination and cross-contamination. So contamination is the introduction of impurities of a chemical or a microbiological nature or of foreign matter into or onto a raw material, intermediate, or an API. For example, it can occur during production, it can occur during sampling of the raw material or sampling of the production during the process. It can occur during packaging or repackaging, as well as during storage or transport of the material offsite. Cross-contamination is the presence in a particular product of small, traceable quantities of other pharmaceutical products manufactured at the same time, in the same premises, previously on the same equipment. And where does cross-contamination originate from, or how does it happen? Well, it happens because of poorly operated and maintained air handling systems and duct extraction systems, which is commonly known as HVAC systems. It happens because of inadequate procedures for personnel and equipment, so inadequate gowning procedures, or people aren't following the gowning procedure correctly. Equipment isn't cleaned correctly either before or after manufacture, and insufficiently cleaned equipment, so equipment used for sampling, for example, isn't cleaned adequately between batches. So the different types of contamination include physical contamination, which is things like particles, fibers, metal parts, anything that is physically can come from the actual manufacturing process. For example, cross-contamination could occur from leakage of oil, for example, from a reactor. A chemical contamination is another one, so that includes things like moisture, gases, molecules, or even vapor. If a product is exposed to a high relative humidity environment, well then this increases the moisture content. For chemical contamination, an example would be when the product is stored in a container that previously held another product but was not properly cleaned. The remaining product's chemical composition may add impurities to the new product, causing it to become contaminated. So for biological contamination, we talk about microorganisms, fungus, bacteria. An obvious example would be improper cleaning of equipment or use of unclean equipment for sampling or unclean equipment during the process. For biological contamination, bacteria may thrive if the container is not properly cleaned and dried. The contaminated container will then affect the product, and microbes may thus be introduced into the batch. So causes of biological contamination include unhygienic and unsanitary practice, improper work attire, so improper gowning, use of contaminated materials and equipment, or if an operator has an open wound or a lesion, that's a source of contamination, or an operator is suffering from a common cough or common cold or some sort of infectious disease, that's going to cause contamination of your batch. And it's important to remember that most raw materials for pharmaceutical products support some form of microbial growth, depending on the nutritive properties and moisture content. So dry powders or tablets are capable of undergoing some form of microbial spoilage or degradation. And the more serious problem of microbial contamination of tablets is where there's no obvious signs of spoilage. Survival of microorganisms within pharmaceutical products is dependent on the particular environment, and it's sometimes influenced by the presence of relatively inert materials. So microbes can be more resistant to heat or desiccation in the presence of starch or gelatin. So they have a longer survival time than in that environment. So contaminants are the presence of anything in the manufactured product which should not be there. It's as simple as contaminants can originate from the environment, it can originate from equipment or from the personnel working in the area. It can come from outside air carrying dust into the site or into the manufacturing area, which is a contaminant. People generate contaminants. So we shed our outer skin every 24 hours. A person walking will liberate 5,000 bacteria per minute, and a single sneeze can produce up to 1 million bacteria. The manufacturing process itself can generate contaminants, for example, paint peeling off equipment or dust coming off off drive belts, things like that. So just to go back to dust again, dust is a carrier and it has to be controlled at all times. Ambient bacteria can be removed by filtration, and internal bacterial distribution can be controlled by directional airflow and air flushing and use of appropriate HVAC systems. Surface bacteria is controlled by adhering to very strict cleaning procedures or standard SOPs. HVAC systems then are heating, ventilation and air conditioning systems used to control your environment. Its purpose really is to provide thermal control and acceptable indoor air quality. These air handling systems are the main tool for reaching required parameters, but they're not sufficient on their own. You need additional measures like appropriate gowning within your area, proper clothing, proper changing rooms, and you need validation of the sanitization of those areas as well. Adequate transfer procedures for materials and personnel within those sterile environments is important also. So this schematic just gives a summary of what we've talked about. So microbial contamination can come from various different sources like equipment. It can come from the process itself. It can come from materials used in the process. Utilities, for example, it can come from the water that's brought in during the manufacturing process. Personnel, inadequate gowning or unsanitary working conditions, and it can come from the facility in relation to the HVAC system, for example. So that's just a visual of what we've already spoken previously about. To summarize again, contamination and cross-contamination are caused by both contaminants coming from equipment and also contaminants coming from the environment and from the operators or the personnel in the area. Cross-contamination is a sure sign of bad practice, as it shows that there is insufficient control over the design of the premises and the system's quality, insufficient control over the air handling and the dust extraction systems, insufficient control over operation and maintenance of the air handling and dust extraction systems. There's inadequate procedures for cleaning of equipment and for restriction of the movement of personnel, and inadequate procedures for the cleaning of the premises. Prevention of contamination, some obvious points would be to determine the cause of the contamination, anticipate the effect of a particular work practice in your area. Is that going to cause contamination? If so, change it, remove it, prevent any ingress or egress of possible contamination to the area, and minimize the effects and quarantine your area. So prevent personnel coming and going from that area if it's not necessary, and control the remaining contamination within your area if it has been contaminated. Again, some points to think about when you're preventing contamination is obviously to eliminate the source. So remove the containment carrier. So human involvement there would be important, because the presence of personnel in the area is a containment carrier in itself. Regulate the use of equipment, because equipment can contribute to contamination, and regulate the use of the air and the water used in the system, in the manufacturing system. Reduce human carrier risk, so proper attire to be worn at all time. Reduce water as a carrier, so water can contain microbes, and waterborne contaminants can be introduced by using water in your process. Reduce air as a carrier, so control your airflow through your air handling units. Use of airlocks is important then between areas. Installation of HEPA filters and ultra-low particulate air will control any dust coming into the area. So tips to prevent contamination again, these are just some pointers in case you ever need to think about these. Some of these might help. So one thing would be to test one material at a time to prevent cross-contamination. Take a sample in a room that has a suitable air-controlled system to prevent contamination through the airflow. Use proper tools designed for the product. Ensure a proper cleaning of equipment to prevent any biological contamination. Regularly check if the cleaning process is effective. Regularly check equipment for wear and tear to prevent any compromise to its integrity. Properly design airflow systems to prevent airflow contamination. Dispensing stations should have proper dust extraction systems, and never return used samples to their original containers. So only take out the amount you need, and if you have extra, you never return that back to the original container. Regularly monitor water to check for presence of microbes. Avoid charging two materials at a time in the production area. Avoid unloading different materials for different batches. Line clearance must be observed during product changeover. And as a guide, we'd often say in the QC lab, total impurities must never exceed 0.5% in your API, and a single individual impurity should never exceed 0.1%. Now, that's just a general rule, and it obviously will change on a case-by-case basis depending on what you're testing, but that's just a general guide on what you should expect in your API samples. So moving on then to clean rooms. So what is a clean room? You may have heard of that or be very familiar with it. It's a clean room is an environment where the particulate contamination and bacterial contamination are limited to prescribed levels. Parameters that need to be controlled within a clean room environment are temperature, air cleanliness, air movement, humidity, room pressure, and lighting. And clean rooms can be classified differently depending on the regulations. So the WHO guidelines would classify clean rooms as either Class A, Class B, Class C, etc. And the United States FDA would classify them as Class 100 or Class 1000. The classification required is dependent on the manufacturing process being carried out at the time. So finally, the regulations around cross-contamination and contamination prevention. In order to prevent cross-contamination from occurring and guarantee the well-being of workers and patients, regulators are scrutinising manufacturing practices, they're tightening manufacturing requirements and around GMP regulations. Some techniques and procedures may vary in different countries, but regulators are still able to come up with globally applicable recommendations and conditions which aim to enhance currently used methods. So the EMA have introduced changes to the regulations, including new points and principles to several chapters and sections. The link is there, again, included in the supplementary material. Please take a read of that when you can. The regulatory authorities request adoption of adequate design and operation of manufacturing facilities, appropriate use of quality risk management, it comes up again, appropriate use of QRM principles is also necessary in order to assess and control the cross-contamination risk that manufactured drugs may actually pose. And that's the last slide. Thanks for listening. And that's the last lesson in this unit, so we can move on to the next unit.